All right. So today is the 14th July 2020, and I'm very pleased to have Philip Askenase, Professor of Medicine and Pathology from Yale University School of Medicine from the USA. So Phil has studied immune and allergic disease model for more than 40 years. He was chief of the allergy and clinical immunology section in the Department of Medicine at Yale Medical School for almost 30 years. He has been founding the AIDS clinic at Yale and participation in the discovery of Lyme disease. So his research has received many awards, including the American Academy of Allergy, Asthma, and Immunology Lifetime Achievement Distinguished Scientist Award. That's such a long sentence in 2017. So his laboratory now is dedicated to study exosomes, particularly as treatment. So it's very interesting today that we're going to learn from him how COVID-19 or why COVID-19 therapy with mesenchymal stromal cells and convalescent plasma should consider exosomes involvement.
Well, I'm very enthused to be able to present to this wonderful group in this fantastic series that you run. I wish I could see the other talk during the week, but of course I can pull it up on YouTube. I'm going to talk today about where our EVs, and I use the term exosomes, might fit in to current biological therapies for the COVID-19 diseases. We're waiting on vaccines, a long way off. We really don't have effective drugs, and it's and so these are two biological approaches that are being maintained. And my message here is right away that the mesenchymal stem cell approach to treatment really should be using their exosomes that are proven already superior to the parental cells. Secondly, turning to convalescent plasma, which is given for the idea that there are protective antibodies that develop in people who have the disease that now after the disease when they're convalescent, using their plasma, the antibodies might be helpful. And it is completely being overlooked that there are literally billions of exosomes in the plasma. If you're giving 500 ml of plasma, that's roughly 500 billion exosomes. And for these opinions of mine, I have examples from our own work. Turning first to the mesenchymal stem cell, which are very healing for a variety of reasons, their exosomes have been shown now incontrovertibly to be the reason that they work. It's been in model systems of injury, like myocardial infarction, stroke, spinal cord injury, and renal, liver, and lung. In autoimmune models, I don't know why my throat is. Lupus, rheumatoid arthritis, type 1 diabetes, and in various CNS diseases, EAE, the model of multiple sclerosis, Alzheimer's, in which, interestingly, nasal exosomes may be helpful, Parkinson's disease, even all the way out to autism. But I would say in the field that cell therapy is slowly replacing exosome therapy, but there's a lot of delay about that. The people in the field have been using the cells for 20 years, and they're a little reluctant to take on our world. And the current plans in COVID are to use the cells and not the exosomes. And that is my point that they're overlooking. Now, there are potential toxicities of using the cells and not the exosomes, because the cells are known to aggregate and on their own can promote coagulopathy with aggregates in vessels, especially the microcirculation. So for the lung dysfunction in COVID-19 patients, there might be a synergy between the vascular injury in the lung and promoting the ARDS, that's acute respiratory distress syndrome. Similarly, these peripheral vascular aggregates might synergize with the vasculopathy of COVID-19. The virus is known to actually directly injure the endothelial cells. So that's, for this particular situation, to me, a bad idea to be using the cells and not the exosomes. There are many favorable properties of the exosomes. They have themselves less innate and acquired immunity. They characteristically don't have MHC class 2 or HLA-DR or other surface co-stimulatory molecules, so they're not really seen very much by the immune system. They also have a surface signature of molecules that together is sort of a license plate or a hashtag for binding similar arrays, coordinating arrays on cells, so that there's an aspect of target cell receptivity that the cells don't have, but that the exosomes do. Further, there's a very long list of problems of using the cells. Imagine you're in the hospital and you want to use the cells, you've got to be culturing them and seeing about their viability and titering them, whereas the exosomes could be on the shelf, ready to go. In our own work, we've studied spinal cord injury, and I want to show you our data that the exosomes from the mesenchymal stromal cells are as good at healing and have allowed us to begin to understand the pathogenesis of how the exosomes might work at the site. So mesenchymal stem cells may not be familiar to everybody, but it's possible to grow them up from starting at only 0.001% at the source. In our case, it's the bone marrow, but there are many sources. And coming up to 100% is 100,000-fold enrichment. So we are dealing with a pharmacological dose and must keep in mind that such a high dose might have unfortunate consequences, but generally they do not at all. You can grow so many because they're plastic adherent, but they're non-phagocytic compared to macrophages, and they're self-renewing with retained and maintained properties. They have a unique phenotype of CD array on their surface, and they lack the typical subset markers of T cells, dendritic cell macrophages, or B cells, and as I mentioned, no MHC class 2. So mesenchymal stem cells are non-T, non-B, non-myeloid, non-macrophage DC, non-NK, INK, and non-embryonic. They are different. So here is an experiment with spinal cord injury in rats. There's a machine that's used, I don't do it, that injures the spinal cord of rats in a homogeneous fashion. And there are 8 rats per group in these studies done at the Veterans Hospital at New Haven that we're collaborating with. And they have a sort of a locomotion index, it's called the BBB score. And they start off here and you injure the spinal cord and it goes down to the bottom. And then slowly, weeks later, there is some spontaneous healing. And if you give a single dose of 1 million mesenchymal stem cells 10 days after the injury, compared to control medium, there is definite healing. That is statistically significant in these groups of 8. There's just this one shot at day 10. This can be quantitated nicely from the inflammation with PET scanning, and you might be able to see that there's sort of less inflammation. But the clearest thing is to look at Evans blue dye, which is given to measure vascular permeability aspect of the injury and inflammation. And this is the dye permeability in an intact spinal cord region. And this is the permeability with the injury. And this is the permeability adding the spinal, the stromal cells, the mesenchymal stromal cells. It's obviously helpful. So there's improved locomotor function, improved lesion size, local vascular permeability after a single dose. Well, if one goes on and gives multiple doses over time, one can get quite a bit of healing. So these are duplicates at baseline. And with the injury, you can see there's this major wipeout of the central part of the spinal cord. But then with repetitive stem cells, one gets this terrific healing. And we're at the point where now patients are beginning to be given the mesenchymal cells, and we hope in the future through collaborators that our Veterans Hospital has in Japan to be able to go on with their exosomes. So we did an experiment to label the mesenchymal stem cells to try and understand their interaction with the spinal cord. And they're labeled such as this bright red, and they end up in the liver, the spleen, and the lungs, and not a one in the spinal cord, absolutely blank. So there's no cells going to the site that is healing. And so we took the supernatant from growing the cells and converted that into exosomes by standard methods. And here are the exosomes. And we're able to do the sizing with a computer program we have for the electron micrographs. And they're in the range of exosomes. When they're given, and that's the red line here, there is definite improvement. And I'm sorry, I've lost the slide of the data that's complete now. That's absolutely the same curve in giving the exosomes as giving the cells. Well, you can see very clearly, here's the vascular permeability with Evans blue of the injury. And this is the conversion of that with the stem cells, and the exosomes are just as good. Then we're able to do 2-photon confocal antibody microscopy at the site. And we have red staining M1 macrophages. They're stained with INOS, and M2 green macrophages stained with CD206. And the exosomes themselves are stained with the lipid dye DIR. So if you follow along, there's a lot of M1 type macrophages, there's a lot of M2 type macrophages. And following this particular M2 macrophage, and looking now just for the exosomes, you can see that there are aggregates of exosomes in the M2 macrophages. So there's a preferential receptivity for a subpopulation of M2 macrophages. Showing that a little further here, this takes us quite a leap because, first of all, going from the cells to the exosomes is a 1,000-fold purification. Now going to these cells and this heterogeneity, we'll be able, with the fluorescence of the exosomes, to isolate the subpopulation of the M2s, which I think will show a subpopulation of the exosomes for us to analyze perhaps what microRNAs and what they're doing to the actual actions of the macrophages that probably influence the inflammation in the tissue. So to summarize this part of things, the literature clearly shows that the exosomes are as good as the stem cells with less toxicity, much more clinically convenient, that there's healing with just a single dose of the exosomes and with multiple doses over time. And lastly, there is a subpopulation of M2 type macrophages that are very receptive. Now I want to move on to convalescent plasma and its use in COVID-19 patients. It is being given, as I mentioned, hopefully for specific antibody content that would be profitable to treat the patient when they're having ongoing active disease of the severe pneumonia and ARDS and cytokine storm as an antiviral and a specific antiviral measure. The convalescent plasma has been given for a lot of diseases, for diseases that are related, and in the SARS epidemic, also MERS and Ebola and avian flu and influenza. The convalescent plasma was given and generally seemed to be helpful, but these were not well-controlled studies. If one looks favorably at them, there's a 50 to 75% improvement, but the results can be very choppy, and there are studies in which there's no effect. And last week, there was an excellent study from the group in the Netherlands who seem to know what they're doing, and they had no effect. And curiously, the antibody levels in the patients when they got the convalescent plasma was the same level as the convalescent plasma. So there's a lot to understand. But there have been very, very few studies that have recognized the role of the exosomes. And now we have thousands of patients that are going to be treated with convalescent plasma, and I'm urging them to consider the exosomes. There's a long history of convalescent plasma. In the early 20th century, it was given for viral poliomyelitis and mumps and measles. Even the Spanish flu epidemic used it. The Nobel Prize was won in 1902 by a noted investigator who gave it for a case of tetanus, realizing that the convalescent plasma of somebody that had tetanus might be helpful. It was helpful, and it established convalescent plasma as an approach and resulted in the Nobel Prize in Medicine. Before antibiotics in the 1930s and 40s, convalescent plasma was used, or the serum of animals immunized with specific organisms was used as the only organ-specific mode of treatment of infections like bacterial pneumonias before we had antibiotics. There are certainly unusual properties of the exosomes in the plasma. First of all, there's billions of them, as I said, and subpopulations of them have tremendous resistance to noxious environment and can survive an acid enzyme hypoxic environment, as will be found in the destroying lesions in the lungs. As part of a viral illness, the virus stimulates by itself cells to produce various exosomes and to trigger the immune system to produce exosomes. And it's likely during the illness that there are pro-disease exosomes. And it seems possible that in the convalescent plasma, there is convalescent exosomes. And they might be targeting M2 macrophages, also Treg cells that can make a lot of inhibitory cytokines, also make suppressive exosomes. And it would seem to me the most optimal therapy would to pre-select plasma for the best antibodies and the best exosomes, not just the mere antibody titer. But there's very little known about exosomes in COVID-19. As I mentioned, during disease, they would be pro-disease and later, hopefully, anti-disease. But they could have negative effects. In cancer, you know, as effector, T cells are suppressed by PD-L1 positive exosomes. And there's a growing knowledge that in an infectious disease, the same process can happen. In Ebola, it was shown that their exosomes had viral suppressors of T cells, and that could be going on here. So the list could grow and grow. Now, the experimental work we did, an early part of my career, I was in infectious disease immunology, and we noticed in patients with certain chronic infectious diseases like viral diseases and fungal diseases, that there was a suppression of the T cell immunity in general. That is, skin testing with tuberculin or with extracts of fungi and other things to which they normally would have a cold delayed type hypersensitivity reaction were suppressed. And so we set up a model, first in guinea pigs and later in mice, of just the antigen part of it, of not studying the viral infection, but of studying the immune aspect, that is, of chronically giving a lot of antigen, which was suppressive, harvesting the convalescent plasma, and testing its effect on an in vivo T cell reaction. We found that it was suppressive, and it was the exosomes in the convalescent plasma was responsible, and we processed to that and tested further. Now, the test we use is currently with ovalbumin antigen immunized mice that get a high dose and then a low dose test injected into their ears. And 24 hours later, there's a lot of swelling of affected T cell cytokine inflammation and a very substantial increase in the thickness of the ears I'm going to show you. So in order to test the convalescent plasma, we immunize the mice as were, but harvest the effector T cell antigen presenting cell mixture and pulse it with the convalescent plasma, or eventually the convalescent plasma exosomes or the exosomes from lymphoid cells, then wash and transfer the cells, altered or not, to naive recipients intravenously and test their ears with a low dose. And we evaluate the suppression of the DTH response. So here at last is the convalescent plasma in this sort of viral-like antigen exposure model. The positive ear swelling here in units of micrometers, 10 to the minus 3 centimeters, is here. And down here is the convalescent plasma, and there's a tremendous reduction and 81% suppression of the ear thickness. Controls here are the exosomes from normal plasma or from sham donor plasma. That is, mice that are treated exactly the way I showed we do, but without the antigen. And in both instances. And you can note also that the supernatant of lymphoid cells has the same exosome activity. Here's what it looks like at the ears. This is the thickness of a mouse ear that is a negative control challenged with the same antigen. It's not irritating. And this is the challenge of the immune. It's an enormous inflammatory response. This is the cartilage down the middle of the ear, and this is the epidermis. Usually there's much more swelling on the side of the vessels. And you can see there's a lot to measure. In the case of the cells that were transferred, that were pulsed with the convalescent plasma and then the convalescent exosomes, there's a definite suppression. Here it is quantitatively over in the lower left-hand corner are the controls, the swelling in the positive, and the suppressed. Now, there's another way to test the exosomes, and that is to inject them directly into mice with these responses. So in this case, they're immunized and they're challenged, and you wait till 24 hours at the height of the inflammation, and then intravenously inject the exosomes and measure the swelling of the ears over time as an indicator of the suppression. And here is a remarkable result. So at 24 hours, they're receiving the exosomes. The dose is 5 times 10 to the 10, roughly a physiological dose. And the intravenous is suppressive, but not terrific in this case. IP is better ID, but remarkably orally administered exosomes are perhaps the best. And this is a repeatable result and it's going to keep us busy. And it has certainly great clinical relevance that oral exosomes and this has implications for other aspects of our field. Now, a peculiarity of our system is antigen specificity. We published in 2013 that exosomes could be antigen specific due to light chains and not heavy chains or whole immunoglobulin on the surface, and nobody has confirmed that yet. And I hope they can. We keep confirming it. So these are the T-cell derived exosomes, and they're positive for typical tetraspanin, CD63, CD9, and CD81, and for kappa light chains. And with dual staining, the CD9 particles are positive for light chains. And there's a mass of them that are low positive and a subset that are quite positive. I'll point out also that the normal controls from non-antigen treated mice have a very low level, so that anybody who tries to do this should be able to detect a low level on normals. And I'd be glad to help anybody do this. Our data from other systems shows that this is a widely occurring phenomenon. We have a lot of publications if you look under my name and light chains. The fact that there's antigen specificity of the exosomes with antibody on the surface allows us to do antigen affinity chromatography to enrich the subpopulation that has the activity. This is a system in which they were, a hapten was, so these are anti-hapten exosomes. The hapten is conjugated to a matrix on the column and goes down, and I recommend this as a technique. It's a bunch of batches instead of one batch. After this batch, you go to the next batch and so on, and you're getting chromatography. And for us, there was a flow through of 90% of the exosomes with no activity, and recovering the ones that stuck had all of the activity. We were able to collaborate with the laboratory at the Rockefeller in New York with cloning, cDNA cloning, and sequencing and bioinformatics hinted that microRNA 150 was involved, and we were able to do biological studies on 150 knockout mice to confirm that 150 was absolutely the microRNA involved, and it has been that in subsequent studies we've done. Recently, we've done anti-CD9 columns, anti-ovalbumin columns, anti-casein columns with the same kinds of results. Now, all in all, in this system, we really are dealing with a circuit. So the high-dose antigen I've been telling you about, stimulate CD8 T cells that are not Treg cells, and they make the exosomes that inherently contain microRNA 150, and also have a binding site or something for the light chains that come from B1A cells and also activated. So we have the first question in our new publications. I want to just give you highlights of trying to answer the question, well, what is the antibody binding? And we found biologically that the target of the exosome is antigen-presenting cells, and now have established that the antibodies are binding to peptides in MHC on the surface, and the alteration depends on the 150 delivery to induce them to make secondary exosomes from the presenting cell that have peptide MHC on their surface, and they bind the conjugate fashion to the T cell receptor and alter the T cell for suppression that can be measured in various ways. So I want to show you just very briefly what we have about this new data that it's the binding the peptide, and this very interesting work at the immune synapse. So these are monoclonal anti-ovalbumin light chains, 2 different ones. We recently will be publishing a paper with 4 different ones. And this is the transferred reaction. These are transferred into DTH T cells alone. And in this case, they are being pulsed with exosomes from tolerized mice that can't make antibodies. They're JH negative mice, but they can make exosomes with microRNA 150, but without the antibody, they're not suppressive. But now we add the antibody to those exosomes from tolerized mice that don't have antibodies, and they're very suppressive, 99% suppression. With this antibody, 93% suppression. And then if we use ovalbumin peptide mixing for blocking, there's very, very strong blocking. So this is one of 4 different approaches we've had to establish that the target of the antibodies on the exosomes is the peptide of the antigen on the surface of the presenting cell. I haven't said much about microRNA 150, but in our system, unusually we are able to incubate the activated exosomes with 150 itself and do dose response experiments and find that very, very little microRNA 150 is sufficient. We even are able to reconstruct the system, given what I've said, to use exosomes from B cells that are antigen immune B cells that have the antigen specificity on their surface, but aren't carrying 150. But if we supplement them 150, this is no exosomes. This is the B cell exosomes as is, added to the T cells, no suppression, but adding the micro 150 to the antigen specific B cell exosome causes suppression. The last thing is what I feel very exciting work, and we're able to transfer the system to a clonal human system with the help of the laboratory of Dr. Sanchez in Madrid, with who we've entered a very profitable collaboration. One can use the mouse exosomes in the human system because microRNA 150 is exactly the same in mice and humans, and the B cells, which here are the presenting cell, can be pulsed to express ovalbumin peptides in MHC, and the antibody, of course, of the exosomes is binding just to the peptide. Under normal circumstances, this classical synapse of the presenting cell and the T cell, without the treatment I'm about to tell you about, generally leads to a positive T cell response. Again, with the diagram, however, if we pre-treat with the exosomes that, as I said, are anti-ovalbumin, but are carrying the 150, and they actually treating these the B cell antigen presenting cell. Then there is a dramatic changes I'll show you in a moment by this ultramicroscopy of the formation of exosomes in the multivesicular body that themselves are suppressive and wind up in the T cells and produced in the environment and with suppression elsewhere. So this whole suppressive T cell response depends on the exosomes transferring 150. If they're 150 negative exosomes, one does not get these suppressive, secondary suppressive exosomes. So here we are with the two photon confocal antibody. The CD81 in the B cells is marked by fusion with a gene of green fluorescent protein. So all this is CD81. And the CD3 of the T cell is marked by antibodies to CD3. And characteristically, at an immune synapse, which is here, there is a migration of the T cells to the point of the synapse. And this is the Jurkat T cell. Incubation for just 2 hours has caused this dramatic change. And this is a multivesicular body of the B cells, full of exosomes that are CD81 positive. And again, you've got the T cell receptor to see the lineup. And then by 24 hours, some of these exosomes are actually in the T cell to influence its activity, perhaps production of interferon gamma and other aspects. So here we have at a single cell human clonal level showing that the murine antibody-coated exosomes delivering microRNA 150 have altered the presenting cells to fuse with the T cells and be activated to produce exosomes that are carrying a different microRNA that have peptide MHC on their surface that then allows this transfer and suppression of the T cells. And in addition, there is release of these exosomes, and they have suppressive capacity. Here we have a murine monoclonal system. It's a so-called OT2 mouse in which there has a transgene of a T cell, single T cell receptor, in which known that the peptide, ovalbumin peptide 323, is the specificity in MHC of the T cell receptor. And if we do our thing here, these are the delayed sensitivity in this particular mouse strain, very, very vigorous. And if we treat the immune cells with the secondary exosomes I just showed you forming in the a multivesicular body, there is a substantial suppression, 41%. And then very interestingly, and perhaps relevant to the subject of today, if we add the anti-peptide antibody to the suppressing exosomes, we augment the suppression. So we have here a synergy between convalescent antibody and convalescent exosomes suppressing this well-known T cell response. So in summary from what I've said today, I think it's a major oversight to use the mesenchymal stem cells instead of their exosomes. That convalescent plasma has been used for more than 100 years and is to be used here in many studies of the efficacy. But now we know for the first time that convalescent plasma contains exosomes at something like 10 to the 10 per mil. This was not known previously, and no one has previously done anything about it. The aim is all on the antibodies, but I feel it's likely that the contained COVID convalescent exosomes in the convalescent plasma participates in the therapy at some level. Maybe they're inhibitory and they should be removed. Maybe they're positive and that should be taken into consideration in choosing the antibody level. This work has been going on for some years, and many wonderful collaborators at Yale. The spinal cord injuries done at our Veterans Hospital by Jeff Kosis and Karen Lanford. Almost all the experiments have been done with a group at the Jagiellonian University in Krakow, Poland by Krzysztof Paniewski and Katja Nasmik and the late Vlad Tak. And we've had Frank Redigold in Utrecht has been a terrific source of beautifully purified light chains. And finally, the very nice T-cell, B-cell responses I showed you with Raji cells and the T-cells was done with our people bringing the mouse exosomes to Madrid to work with the laboratory of Francisco Sanchez Madrid. Thank you. I'd love to have questions. Do we have time? We do.
Yes, we do. Phil, thank you. Thanks for your wonderful presentation. So let's get on with the questions now. And there are already a few questions which has been posted. First one is from Mike. Mike, can you unmute your, I'll help you with, yeah.
Is that Mike O'Neill?
Yeah, carry on.
Hi, Mike.
Michael, you need to.
I can't hear you.
Mike, you need to unmute.
Can you hear me now?
Awesome.
Yes.
Well, I just wanted to ask if I'm sure you know probably about what they're doing at Rockefeller with the so-called elite responders, and
Yes, I do.
And do you know how that's going, or?
Well, I know there's very, very good people. It's Michel Nussenzweig. I used to work with his parents, who were both fantastic immunology people. And they've taken that to a wonderful point with HIV to find so-called elite antibodies, antibodies that are especially positive and react with, but I know they're doing it, but I haven't heard where they are.
Okay.
But that certainly would be a terrific convalescent antibody to use.
Well, thanks.
Okay, so the next question is pretty much from me. So I'm going to ask that. Phil, that's a wonderful talk. Thanks for presenting some unpublished data that has been gathered from your lab. You showed some data on the CP, the convalescent plasma, and also in terms of showing their usefulness in therapy historically as well. So I'm just wondering, when you're talking about what fraction of the convalescent plasma is functional? Have you guys tried depleting EVs or exosomes from the convalescent plasma and then injecting those convalescent plasma without the EVs into the mice models that you have and see what fraction is functional in that particular plasma?
Well, I'm not myself doing it. There's two very large studies going on in the States. One at Mount Sinai Hospital in New York, which was a huge center when New York was really hot. And they've already published that the safety of the convalescent plasma. And then there's a FDA NIH study that's focally at the Mayo Clinic, involving something like 50 centers in the States and thousands of patients who are going to get convalescent plasma. I spoke to them and they said, well, yeah, exosomes, we kind of know about them, but we don't have time to deal with them. So all of that has to be done. In the view of this study we just had where the convalescent plasma didn't work, I'd certainly, the first thing, they have no negative control. They have antibody. I don't know if you've seen this study. Antibody measured in a certain way. And I think we have to be very careful that the way we measure the antibody may have nothing to do with the actions of the antibody in vivo. I'm a big in vivo veritas person. But they had no negative antibodies. Everybody was positive. They need to go in the freezer and get some pre-COVID serum and plasma out and see if this is a general by this assay. So there's just very little happening so far about the exosomes. To me, the thinking is like from the 30s and 40s. Does the plasma work? And now, you know, we know that antibodies are very complicated. And the immune system has so many different aspects. And now there's the exosomes to consider.
Okay, thanks, Phil. So, thank you. The next question is from Kalyani Patil.
Yes, I'm here.
Hi.
Thank you, Dr. Philip. Your talk is therapeutically very relevant, and the great need for considering the COVID-19 situation currently. So I have basically two questions here. So when you derive or when you obtain the convalescent plasma from the COVID-19 patients, so since we don't know much about the exosomes in the convalescent plasma, so do you think that you would obtain a mixture of exosomes containing partly some exosomes would be containing immunosuppressive cargo, and partly some exosomes might be containing the antibodies? So
Yeah,
how would that work then?
I think at a minimum that's true. We have tremendous diversity in them. I showed this flow cytometry where all the exosomes are positive, the CD9 and for the antibody, but you put them down an antigen column and 90% come through. So there's 10% that have characteristics that allow them to bind the column that have the activity. So yeah, there's, you know, Clotilde wants me to say EVs, and she's right, but I'm stuck with exosomes. There's tremendous diversity. You certainly could have a mixture of positive and negative acting exosomes, and a column might separate them. Yes.
Okay. Okay.
Please,
My second question.
Please do the experiments. There's a lot of convalescent plasma where you are.
I mean, I don't know. I'm not in India currently. So maybe our population is less here in Qatar. So, but yeah, I do like to mention here that the Qatari government has started administering the convalescent plasma in the patients here. So they have already started with the plasma therapy.
Wonderful, wonderful.
Yes. And my second question is, you were mentioning about the MSC derived exosomes as well as the convalescent plasma derived exosomes. So from your experiments, it's clear that the convalescent plasma derived exosomes are CD9 positive. And we also know that the MSC derived exosomes are also CD9 positive. I happened to read one paper which was published just a month back or so, that CD9 actually helps in virus docking together with ACE2. So I'm quite confused here that the CD9 that is expressed on the exosomes, is it underexpressed, or is it, I mean, structurally modified? I mean, it's still expressed, but is it structurally modified?
Well, besides the receptor interaction about the angiotensin enzyme, there's a peptidase that is also needed. And the CD9 is new information. And certainly what you're saying has to be considered. And it might lead to positives and negatives. And yeah, I mean, we don't really know a lot about what the role. Margaret Zoller has been working forever about the action of the tetraspanins, and they've got to be doing important things. But I don't think it's nailed down very well, and that's a very interesting aspect, that it has to do with perhaps pathogenesis of viral illnesses. Send me the paper, please.
Oh, sure. I would.
Thanks.
Thank you, Dr. Philip. Thank you.
Okay, so the next question is from Abhimanyu Thakur. Abhimanyu? Yeah, he's online.
Yeah. Hi, Philip. It's a nice talk, actually.
Thank you.
Thanks. So I was just wondering, like, whether this SARS-CoV-2, whether it is present in exosomes, and is there any, like, can it even cross this, like, blood-brain barrier? Because I'm currently working in neuroscience and trying to explore, like, whether like this SARS-CoV-2, whether it is, like, it can be present, because the size are almost similar. So can you give your some comment over this?
Well, there's growing information that the viral infection involves neurological aspects. How that happens, I'm not sure. I think you know that it's known that exosomes cross the blood-brain barrier with no problem. I have a paper that's posted in preprints all about the advantages of exosomes compared to artificial liposomal that don't cross the blood-brain barrier. So certainly, if exosomes are there and are important, they could be crossing the blood-brain barrier, perhaps contributing to the neurological illness or antagonizing it. But certainly should be considered. Definitely, it's a very, very interesting and important area. I wish you'd pursue it.
Thanks, Philip. Thanks.
Thank you.
Okay, so next question. Andy Flies, do you have a question or?
Yeah, thank you. Thanks for the talk, Phil. Curious about the role of intravenous immunoglobulin for treating systemic lupus erythematosus. As you mentioned that people are attributing things to antibodies usually, and IgIV is used for lupus. And I'm wondering if anybody's looked at exosomes for that. And also following up, kind of part two of the question is if exosomes are expressing light chains, any chance that those exosomes could react with an FC receptor? Because I think the IgIV treatment for lupus is targeted at antibodies and FC receptors. So having a light chain in the mix could be pretty interesting.
Well, first of all, the IVIG doesn't have very many exosomes in it. It's been processed a lot. We give a lot of it in a lot of diseases. As far as FC goes, light chains don't depend on an FC receptor. And we did the experiments with FC receptor knockouts and shown the exosomes were perfectly, well, the light chain biology was perfectly all right in FC negative. So I'm not sure I'm answering all your question though.
Yeah, I think you got it. I was thinking more along the lines that if the so some of the purification systems, so not protein A or G, but one of the newer ones, the L, seem to bind some of the light chain a little bit. So I'm just wondering if you have a light chain there, and there's even a small affinity that if you had an exosome with some other proteins, that the avidity.
Yeah, I hear you.
Increase.
I don't know if IVIG contains light chains or not. That's a pretty fundamental question I don't know the answer to.
Thanks.
I mean, it's pretty much the talk. We just had a talk last week about TAM horseful protein and the urinary exosomes. It was a terrific talk at maybe the other talk, and that has an affinity for light chains, and it's the major protein in urine. And in myeloma, which is a tumor of plasma cells, they make a lot of light chains. And light chains are made normally by plasma cells. And in myeloma, where there's a great deal made. There are precipitates of the light chain with the TAM protein that tend to shut down the kidney as a major problem in myeloma. I don't know about FC. I mean, I actually started life on FC receptors bearing immunoglobulins. So I think I'd love to talk about it with you in email. So
Yes.
send me an email and let's discuss it.
Sounds good. I have your email from the weekend about the recent paper, so I'll follow up.
Do you work with these little creatures that these have this viral tumor?
Yeah, I work with the Tasmanian devil facial tumor mostly, but I cross a lot of boundaries.
Very, very interesting model. Wow, yeah. Love to talk to you about it.
We'll follow up. Thanks.
Thanks, Andy, and thanks, Phil. So if anyone has any last questions for Phil? If not, let's thank Phil for a wonderful presentation on a topic that is very, very persistent right now. And also, thank you all for your attendance and being patient throughout and asking wonderful questions. Thank you.
Okay.
Well, we'll see you all at the meeting that's coming up in the next 10 days or so. And it's a very exciting preparation going on for that.
Yeah, so this Thursday will be our last WebEVTalk before ISAF 2020. So it will be an interesting talk on MSC, extracellular vesicles with Bernd Giebel. So if you would like to join us for the Europe session, and we'll have but a couple of weeks break and we'll return to WebEVTalk after the ISAF 2020. And I hope everyone joining the meeting, the ISAF 2020 is going to be very exciting and might be the biggest attendances yet. So see you soon. Yeah, and yes, and stay safe. And thanks so much, Suresh. It was great.
Thanks, guys.
Yeah, thank you for coming, Suresh. Thank you.
Thanks, guys, all of you.
Okay, bye, everybody. Thank you so much for your comments.